This is a video documentation of Team X1's microfluidics experiment. The goal of our team's experiment was to research if changing the pH of a yeast cell solution would affect the cell adhesion of the cells onto the PDMS. First, we needed to design a chip for our experiment. We designed our custom chip to improve off of the standard chip, which had chip overhang. We added space between the fastener and chip, used identical channels, and removed unused holes in the chip holder. Important considerations that should be taken while designing the chip is to try to place fasteners so that equal pressure is applied throughout the chip. It's important to make sure inlet and outlet ports are large enough to support flow. Also, to prevent leakage into other channels or outside the chip, space the channels generously apart from each other. After designing the chip, an acrylic wafer of our chip was manufactured by the TAs. Afterwards, we obtained 2 petri dishes. We placed the wafer pattern side up into one of the dishes. Next, we drew out a specific amount of PDMS base agent. Then, a smaller amount of the PDMS curing agent was drawn up. The specific amounts of each were calculated using a given 10 to 1.05 ratio by mass, with a total of 15.9 grams. Next, we stirred the solution for 5 minutes, ensuring that the PDMS base was completely cured. Then, we carefully poured half of the PDMS into the dish with the wafer, and then other half into the other petri dish. As you will notice, there will be many bubbles which are in the PDMS. The next step was to get rid of these air bubbles through desiccation. By placing the petri dishes into a desiccator which created a vacuum, bubbles rose to the top of the chip due to difference in air pressure. Then by slowly reopening the valve and equalizing the pressure, the bubbles at the top pop. This procedure of closing the valve, waiting a minute, and then opening the valve was repeated numerous times until most of the bubbles were popped. Finally, the remaining bubbles, or large bubbles that couldn't be desiccated, were popped by hand. After the chip was bubble-free, it was labeled and placed into the oven, where it was baked by the TAs. After the chip was baked, the next step was to remove the chip from the petri dishes. This was done by first cutting around the chip and carefully breaking off the petri dish. Next, inlet and outlet holes were punched for the 4 channels. Finally, the chip was placed into the sonic cleaner and dried off using compressed air. Calibration trials were run using our chip. Before each trial, the chip was cleaned using the sonic cleaner and dried off, and the entire assembly was put together. The calibration trials were run at heights from 50 to 55 centimeters based off our initial experiments with the standard chip. These trials were to see which channel produced the most consistent flow rate, and also as a baseline to compare the flow rates from the yeast cells. The amount of water mass that was in the cup after each trial determined the flow rate. For our yeast cell trials, 2 grams of yeast were measured out on a balance. Then 10 milliliters of a warm salt solution was added to the yeast. The yeast was mixed thoroughly and allowed to incubate for 20 minutes. Finally, the yeast was seeded into the chip using a pipette. There, the yeast was allowed to adhere for another 20 minutes before the trial was run. Using the same experimental setup as the calibration trials, 5 controlled trials were run. During each trial, the team decided to use a low-pressure flush for 22.8 centimeters for 3 minutes. Then, shearing trials, which moved the water height up 2 centimeters, were run for 2 minutes. The height was increased for each trial until significant shearing, which the team defined as 70% of the low-pressure flush monolayer cells, were gone. The treatment trials were conducted the same way as the yeast trials, except the pH of the solution used to incubate the yeast was changed. Taking 1 milliliter of 5% acetic acid and adding it to 10 milliliters of the salt solution, mixing the solution and taking 10 milliliters of the new solution, the yeast cells were incubated in the solution with around a pH of 3. After the low-pressure flush and shearing for each trial, microscope pictures were taken of the cells left in the channel. These pictures were then cropped to the same dimensions so that the MATLAB program could count them. After counting the cells and comparing them to the shear stresses calculating using the flow rate, the shear stress needed to shear 70% of the cells was determined. A two-tailed t-test of the shear stresses between the control and the treatment trials was conducted, and using a 95% confidence interval, we were able to reject our null hypothesis that there was no difference between the standard yeast cells and the pH-treated yeast cells. This experiment was very frustrating at times. The initial heights of 55 centimeters were much higher than the heights needed for monolayer for the low-pressure flush. Many trials were run where the cells could be completely sheared off. The heights were later moved to 30 centimeters and then 22.8 centimeters for the low-pressure flush. Other times, getting flow to the chip was near impossible, and until the green tips were replaced and recut. It's important that through experimentation you remain calm and start early. Eventually, we figured out how to efficiently get data points, but it took over 20 hours in open lab before our first point.